The female reproductive system functions as an interconnected network responsible for gamete production, fertilization, pregnancy, and childbirth. This system operates on the principle of anatomical continuity, serving as a synchronized structural pathway. Understanding this pathway requires looking past individual shapes and focusing on a sequential physiological timeline regulated by hormones. Normal micro and macro anatomy provides the first principal basis for predicting and diagnosing reproductive pathologies. We will trace the exact path of an ovum from its generation in the internal organs outward through the structural layers. Recognizing how these specialized organs integrate reveals how minor anatomical disruptions trigger systemic clinical consequences. This map of the reproductive tract identifies the starting point of the pathway, the ovaries. These paired almond-shaped organs are situated within the pelvic cavity. The ovarian and suspensory ligaments provide the mechanical stabilization that holds these organs in place. A cross-sectional view reveals two distinct histological layers, the outer cortex and the inner medulla. The cortex contains the follicles and serves as the site for oogenesis, or ova production. The medulla houses the internal blood vessels and nerve supplies. Beyond gamete production, the ovaries act as an endocrine center, secreting estrogen to develop female characteristics and progesterone to maintain pregnancy. The ovary functions as the master endocrine control center, dictating the structural changes required for the rest of the reproductive pathway. Once an ovum is released, it enters the uterine tubes, which provide the transport mechanism to the uterus. The infundibulum marks the entry point, where finger-like fimbriae sweep the ovum into the tube. The ovum moves into the ampulla, the widest part of the tube, and the primary site of fertilization. The pathway concludes at the isthmus and the intramural part, which connects directly to the uterus. Within the ampulla, gametes meet to initiate fertilization during a specific biological window. This narrow architecture, however, creates conditions for a major clinical correlation: ectopic pregnancy. An ectopic pregnancy occurs when an aberrant implantation event takes place within the narrow fallopian tube rather than the uterus. While the tube is designed for transport, its narrow structure is vulnerable to rupture if implantation occurs prematurely. The uterus is a pear-shaped muscular organ divided into the fundus, the body, and the cervix. We deconstruct the uterine wall into three histological layers: the outer perimetrium, the middle myometrium, and the inner endometrium. The myometrium consists of thick, smooth muscle responsible for contractions during labor. The endometrium is the mucosal lining that provides the site for embryonic implantation and nourishment. This lining either supports a pregnancy or sheds as menstrual flow if fertilization does not occur. These histological layers categorize different uterine pathologies, beginning with uterine fibroids. Uterine fibroids are benign tumors that originate specifically within the smooth muscle tissue of the myometrium. Endometriosis involves a different layer of the uterine wall. In endometriosis, endometrial mucosal tissue grows anomalously on structures outside the uterine cavity, such as the ovaries or tubes. Distinguishing the myometrium from the endometrium provides the diagnostic framework to separate muscular fibroids from mucosal endometriosis. The cervix acts as the structural gateway connecting the uterus to the vagina. Cervical cancer is the primary pathology directly affecting this gateway. This gateway also presents an infectious vulnerability. Pelvic inflammatory disease occurs when pathogens travel upward through the cervix to reach the internal organs. The vagina is a 7 to 10 centimeter fibromuscular canal that facilitates menstrual flow, sexual intercourse, and childbirth. The external genitalia, or vulva, include the mons pubis, labia majora and minora, clitoris, vestibule, and Bartholin glands. These structures provide physical protection for the internal organs and facilitate lubrication. The lower tract must balance its role as a physical barrier with its function as a biological passageway. The system includes the mammary glands as vital accessory organs. Anatomically, these are classified as modified sweat glands. Their internal structure consists of lobules and ducts designed for fluid transport. Their biological function is lactation, triggered following childbirth. This continues the reproductive timeline, providing the physiological handoff required to ensure neonatal survival. The reproductive system operates as a synchronized timeline, where gamete release, transport, fertilization, and implantation occur in a fluid sequence. Production in the ovaries relies on the specific transport architecture of the fallopian tubes, ultimately reaching the specialized environment of the uterus for gestation. This entire network is driven by a continuous biological flow. In this system, anatomical form dictates physiological function. Identifying specific microanatomy, such as the difference between the myometrium and endometrium, is the only way to accurately map clinical vulnerabilities. The female reproductive system functions as a hormone-driven structural timeline, where every organ, layer, and pathway performs a specific task within the reproductive cycle.